So next we move on to our last speaker, Hitesh Mistry from University of Manchester. So he got his PhD from the University of Manchester in applied mathematics and had a postdoc at the University of Dundee. And he is closely working with the clinician and biologist to investigate drug discovery and pharmacokinetics, and then working with diverse healthcare data science. So today, he will be talk about exploring the key clinical empirical observation relevant to adaptive dosing. So please take away.
Okay, can you see my screen okay?
Yes, yes.
Yeah, full screen, okay. Yeah, so thanks again for letting me give a talk today. Hopefully some of you will like it, maybe not all of you. So just a little bit about my background. So I'm a mixed in terms of, I do a lot of work in the pharmaceutical industry and also quite a bit of work generally within the NHS in my local trust, which is the Christie, which is a cancer hospital in Manchester University. I'd like to start with my acknowledgements actually. So there's two people I'd like to sort of acknowledge mainly related to some of the simulation work I'm going to show towards the end of my talk. And that's Leon Ahrens from Manchester University, and also Nicola Melillo, who has quite a strong interest in sort of control theory and generally applying engineering principles to healthcare. So I'm going to start with a quote, and I'm going to use this paper a few times. So some of you may or may not know Doug Altman. If you know a lot about statistical modeling in healthcare.
I think there is a problem. Could you share your screen to switch your screen, I think?
Okay, where's that?
Yeah, we see your presenter load, so just
okay,
share slide only.
So full slide view instead of the presenter view. Ah, okay. So I want to do which one?
Yeah, I think it's, can you try the play? Yeah.
That one? That's what I just did try. I mean, I'm happy just to do it like this, if that's okay, actually. If you can see the screen okay.
I think we don't see your
I'll just,
yeah.
Oh, right. Is that all right?
So you can share the entire desktop, I think.
I think that's good enough, isn't it? Or,
no, I think
I
we
just did, didn't I?
I don't.
I think this isn't this clear enough though? I mean, if you're on screen, so.
Can you try the slide show?
Yeah, normally I can usually adjust which screen is sharing, but I don't seem to see the option.
I think then, yeah, just I think just please go on. I think it seems fine. Yeah.
This is okay. It'll do. I think.
Okay. Try, yeah.
Okay. So yeah, Doug Altman is a professor of sort of biostatistics, medical statistics based at Oxford. And he's done a lot of work in medical research, and he wrote quite a few papers over the years, over the decades. And I'm going to quote bits of this from this paper a few times. And this is essentially a paper that describes the scandal of basically poor medical research. And I guess his first statement is that we need less research, better research, and research done for the right reasons. So we'll come on back to this paper later on. So the first thing, so just a brief outline of the talk. So I'm going to have a quick look at some of the preclinical cancer evolution studies. And then we're going to talk a little bit what I'm actually interested in. So my interests are generally in sort of surrogate biomarkers in general, and thinking about biomarkers and how we can use them in drug development purposes and making dose adjustments, etc. I'm going to talk a little bit about selection bias as well, and then we're going to come back to the concept of surrogate biomarkers again. So regarding preclinical studies, this is a nice paper by Diane Liston and Myrtle Davis. What they did is just a few years ago is they pulled together all the sort of approved drugs in oncology that are given orally. And they had a little look to see, well, what is the free, what we call Cmax? So Cmax here is the maximal concentration that you see over, say, for example, over a 24-hour period. What does this free drug concentration look like? So free drug here means it's not bound to plasma proteins. This is drug that's actually going to work in an active way. So what they learned is that actually this is the distribution of free Cmax. And remember, Cmax is probably the concentration you're likely to see only for a short period of time, right? Just a few minutes. Now, one of the things that you start to see is that for a lot of the approved orally targeted agents, essentially it's rare to see drug concentrations greater than 1 micromolar. That's a Cmax. So then if you think about a 24-hour average, it seems like the concentrations that patients are generally seeing are a much lower level. So resistance in the clinic, you could argue, is probably happening at quite low exposures. So concentrations that are definitely sub-micromolar. So what does this mean? So the reason why these people wrote this paper is that they were trying to sort of encourage the community to start thinking about, well, are you using clinically relevant concentrations when you're doing preclinical studies, regardless of what study you're doing? And this is a critique and a point we try to raise quite often in a lot of projects that I'm involved in. So apologies to Rob. I thought I'd pick his paper, but I could have picked any really, to be fair. And so in this, for example, this paper that Rob described earlier, one of the things I discuss here is how do you create resistance, right? So how do you create resistant clones? Now, the article itself discusses that to generate some of the resistance to CDK inhibitors. So remember what these drugs have. So in the clinic, what we know is that the free Cmax is roughly 15 nanomolar, right? This is quite low. Now, what's interesting is that when people have been doing experiments, they use far higher concentrations than what we're seeing clinically. In this case, 1 to 2 orders of magnitude. So that's one of the first points I wanted to raise. And another drug in that study, for example, the one that you talked about earlier as well, this new 6102, which is a cell cycle inhibitor. So when you look at this drug, actually its affinity for its targets, CDK1 cyclin B, the IC50s, so the amount of drug you need to inhibit by 50%, is roughly 5 and 10 nanomolar. That's actually quite low, right? Whereas in the experiments, people were using sort of tens of micromolar, 20 up to 50 micromolar. Now, this is a quote from a colleague, but I guess it's a quote that I would use too, right? So when I see drug projects, and I run a biotech too, if someone shows me a drug that requires 50 micromolar but to be active, I will essentially show you the door. I'm not interested, right? So this is the point. I think a lot of preclinical experiments have been using doses and drug concentrations which have no clinical relevance, which kind of makes you think about, well, how relevant are the results from a lot of these sort of experimental studies? And this also, it's another question, is there any really preclinical study that has actually used clinically relevant drug concentrations? From a lot of the papers that I've read, including some of the in vivo ones, they haven't even bothered measuring drug levels. And drug levels do vary quite substantially across same sort of species of animals. So the same strain of mouse you're using for your xenograft experiments, you will find at the same dose, you'll get extremely variable levels of drug concentration. So that was the first point I wanted to raise. The next one is a bit of tongue-in-cheek. So adaptive therapy in the clinic, anyone for selection bias. So this is a nice paper. So what the FDA do every so often, so what you're seeing on the right-hand side is essentially a paper from the FDA. And what this paper represents is essentially what's been happening to patients in terms of prognosis based on their response to treatment. And so what the purpose of the paper was to highlight is that essentially when patients do respond, i.e. if tumors do shrink, those patients generally do better in terms of prognosis than those patients whose tumors don't actually shrink to treat, don't have an effect. So patients whose tumors don't shrink. And one of the other interesting things you can get from this is that actually the response rates in general in clinical trials is actually quite low, right? So I've chosen non-small cell lung cancer. That actually doesn't have a great response rate across all the trials that we've seen. And here it's basically approximately 20% across, I think, a decade's worth of clinical trial data in non-small cell lung cancer. Now, this isn't necessarily the case for all tumors, but it does make you think, right, in terms of public health policy about adaptive therapy. So if the point is that you need a drug that actually has an effect on tumors, not many patients actually, so there's not many patients where the actual drug actually does have an effect that you do see a degree of tumor shrinkage that you could potentially use for your adaptive therapy. So if the goal of adaptive therapy is to optimize treatment, basically you want patients who essentially do actually respond to treatment. And if there's only about 20 to 30% of those patients that actually do respond to treatment, and these patients will tend to do well anyway, as we well know, from a public health perspective, does it make sense to invest time and resource on the 20 to 30% that do well, or the 70 to 80% that don't? So that's an interesting public health perception of adaptive therapy. So when you talk to certain public health experts, this is what they may well ask you. So what am I interested in? So my interest is generally in thinking about surrogate biomarkers. So So I'm going to try and explain this hopefully reasonably well. So my view is that if you want to use a biomarker to make dose adjustments, you better be pretty damn sure that that biomarker does actually fully capture the dose or the exposure effect on the clinical endpoint of interest. So what I've drawn here is a diagram where you have dose or exposure. So this is your drug. Now we know that it does have an effect on an endpoint, hence it was approved. So it does have an effect on overall survival. What I'm looking for is a biomarker which fully captures this effect of the dose effect on survival. If I can find such a biomarker, that would essentially be considered a surrogate, right? So I can use that as a surrogate endpoint. If it does fully capture the dose effect, then if I change the dose, that should hopefully change the biomarker in a way that also has an impact on the endpoint. Now, the point is that actually surrogate biomarkers don't actually exist. They never have done, really. There's one example where there's probably a hint of one, but in general, we don't really have surrogate biomarkers. So then it just changes the question. So how much of the drug effect does a biomarker you want to use to play around with dose have to capture such that your dosing decisions don't lead to some sort of reduced efficacy? So the biomarker here could be many things. So we've heard a lot of talks about PSA, so prostate-specific antigen. It could be imaging variables. It can even actually be another time to event variable, right? So it could be radiological progression itself, if your final endpoint was overall survival. So let's now consider the adaptive dosing therapy trial, one that we've seen presentation on in the plenary, I believe. So my understanding is, so what I look for in metastatic castrate-resistant prostate cancer, and what these studies were powered to look at, is an improvement in overall survival, right? So the key endpoint was overall survival, not PSA progression, not radiological progression, but actual overall survival. Now, in the actual adaptive therapy study that we've heard so much about, there's certain things that are different between that trial and the trials that people have been comparing the results to in the actual paper. So one of the things is in the actual phase 3 trial, patients actually I've never had abiraterone before, right? So the point is that you're not selecting out patients based on their response to abiraterone. Now, in the actual adaptive therapy study, you kind of need patients to have a response first. So in that study, essentially what you've got is your patients are actually patients that initially see a 50% fall in PSA. Now, that's not the case in the actual original trial, right? So in that paper, when people are making comparisons to the original trial, they are biased in favor of adaptive therapy, in the sense that actually you're picking up patients who actually do have a response to abiraterone, the drug of interest, which is not the case in the phase 3 study. Now, the other thing that's of interest here is that we've heard a lot of presentations of people presenting a control arm. Now, what's interesting is that actually in that actual adaptive therapy trial, there's no mention of a control arm in the actual trial protocol, which is available on clinicaltrials.gov. Now, this is kind of important, and I'll explain why. So I'm going to try and explain a little bit about this sort of map here, and also surrogate biomarkers again. Now, going back to the concept of surrogate biomarkers, I'm going backwards and forwards a bit here. So we talked about PSA and radiological progression and not actual true surrogates. Now, what's interesting with abiraterone, there was a study that was done using a lot of the phase 3 trial data, where actually what they found is that circulating tumor cell counts are a better dynamic biomarker than PSA. So their argument would be is actually if you're going to make dosing decisions, you shouldn't be making dosing decisions on PSA, because PSA doesn't actually capture the treatment effect on the endpoint, but circulating tumor cell counts do. Now, this is kind of important, and we'll go on to explain why in the next slide. So one of the things we know about the drug that's being used for describing a lot of these adaptive therapy studies in this one paper is that drug abiraterone actually does have what we know are known prognostic factors. So in the actual phase 3 trial, if you were to look into that paper and actually the clinical pharmacology label, is you'll find that things like age, lactate dehydrogenase, hemoglobin, alkaline phosphatase, and sites of metastases, they're all prognostic factors. So what does that mean? Well, they will probably have an independent effect on the endpoints of interest, regardless of dose and exposure. So what does that mean when you want to compare to historical data? So if you're presented data where you're seeing, for example, a survival curve showing a difference between adaptive therapy and what they call sort of maximum tolerated dose therapy or the control situation, what you want to see is there anything different about the prognostic factors? Because those sorts of differences you're seeing can actually easily be explained away by the fact that the prognostic factors are different between the two arms of a trial or the two parts of a trial. Now, in order to be able to adjust for these things, you kind of have to build a model where you have a regression coefficient that's actually linked to each of these sort of prognostic factors. And there's quite a few. You can't do that on a small study. You cannot do that on a study where all you've got is the order of 10 to 20 or even 30 patients. There's just not enough patients to be able to really make that adjustment to see what the true effect is. That's why I'd always be very skeptical of conclusions that are essentially from an observational studies, which is essentially what you could describe that adaptive therapy study that we've heard a lot about. And what's interesting when you start to dig around in literature actually around about PSA, so we do see PSA being used a lot for modeling studies. And that's also true actually in the PKPD sort of modeling community. And the reason for that is probably because it's cheap, it's easy to measure, and there's lots of it, right? It's time series data, it's easy to get. However, what people have shown actually, it's not a very good biomarker in general for many reasons. And here's another reason. So this is a nice study that was done by David Verbel, and essentially what they found in this sort of cohort of patients they were interested in, they were sort of asking the question, well, if I use PSA as a time-dependent covariate in a survival model, how much of the survival variance does PSA actually explain? Well, it doesn't explain a lot. It only explains around 17% of the survival variance. And that's because a lot of those prognostic biomarkers that I mentioned before actually do contribute quite a lot to patient outcome, much more so than you think actually. And I guess one of the points that they make is that if the consideration of PSA alone as a surrogate can produce misleading information regarding the risk of death, its use as a surrogate biomarker for survival is not warranted when designing a clinical trial. So we've known for a long time actually that most biomarkers only really ever explain a modest amount of the survival variance. And actually we've known this for decades. So this now brings us on to the next part of the talk I want to go through, which is basically a series of clinical observations based on lots of literature data and analysis done essentially by the statistical modeling community. So one of the first things that they've actually noticed is that in many sort of trial data sets is that the more you shrink tumors, the longer the patients live. So if you can shrink it as much as you can, the more you shrink it, the longer the patient lives. However, the link between tumor shrinkage and the endpoint is not drug independent. So there seems to be a drug dependency in terms of the quantitative link between the amount you shrink a tumor and the survival times of patients. So that was the first observation I wanted to sort of present. Another one people have seen is actually when people have analyzed a lot of sort of tumor burden time series data, a lot of papers have also found that there's actually no correlation between how fast you shrink a tumor and how quickly the tumor grows back. So that's interesting in itself. Now, the third one, which is of interest to me, is that what we've also known is that tumor burden is essentially a time-dependent prognostic factor. So what does that mean? So in terms of these survival models, what we're interested in is the probability of a patient experiencing an event. This could be disease progression or death in a certain time interval, and that's usually conditional on surviving up until that time window, right? And what they found is that the probability of surviving up until that time window is actually dependent on the level of tumor burden leading up to that time window. And this is true of many different markers you want to think about, whether you think about imaging data, some of these sort of serum biomarkers like PSA, et cetera, et cetera. Now, before that, I basically said, well, actually, a lot of these sort of biomarkers don't really explain that much of the survival variance. So, but let's just assume we do have this perfect biomarker. And let's also consider this idea that these tumor burden biomarkers actually are time-dependent covariates of the endpoints of interest. So what does that mean? So what is a time-dependent predictor of sort of progression-free survival and overall survival? So a lot of you do use sort of Cox proportional hazards models. I've seen that in quite a few papers that people from this community presented. So I'm kind of assuming you know a little bit about sort of survival analysis. So what have people found? So what are those papers that I cited? What did they show? So they basically took what you call a Cox proportional hazards model or essentially a semi-parametric sort of log linear model. And what they've shown is that the sort of hazard, so the instantaneous risk of death is actually dependent on the level of tumor burden over time. That's what TB of T is, right? So this is the tumor burden over time of an individual patient. And essentially what they found is that there's a positive association, so a positive correlation between tumor burden size over time and whether you're going to experience an event within a given time interval. So let's now consider an individual patient. And just one comment here is that we normally specify this as the hazard of time t is equal to the baseline hazard, which is essentially can be specified as a function, and this function can essentially take numerous distributions, including exponential, etc., and that point's kind of irrelevant. But the main point here is that actually the probability of seeing an event is actually directly proportional to the level of tumor burden at a given point in time. So what does that mean? So if you were to take some of the time series that we've seen that's been presented, so if you think of the left-hand side here, where the y-axis is tumor burden, x-axis is time, and what you're seeing here, for example, is a simulation that looks at adaptive therapy. So the actual dynamical model is kind of irrelevant here when you're linking, because what we care about is a signal that feeds into the time to event model, not necessarily what model actually produces that dynamic itself. So what you're seeing here is the typical things that we see for adaptive therapy. So if you look at the green line, we're essentially seeing that tumor burden is being controlled. Whereas if you apply continuous therapy, we find that, okay, there is an initial benefit, but eventually if you look over a longer period of time, the benefit is seen for adaptive therapy. However, like I said, what we know is that tumor burden is a time-dependent covariate. So essentially what you can do is convert that y-axis to essentially be equivalent to the probability of experiencing an event. Now, if you then now think about the y-axis in that context, actually things start to change. So if you then imagine that actually the y-axis is the probability of experiencing an event, so this could be a death event, then essentially the adaptive therapy, what is it doing? So initially you're reducing your chance of seeing an event, and then as you come off therapy, you're now increasing the chance of experiencing an event. Whereas in that same sort of time period window, under continuous therapy, you're continuously reducing the probability of that patient experiencing an event. What does that mean when we look at a survival curve for that individual? So this is survival probability over time for an individual on continuous therapy versus adaptive therapy. Actually, what you find is that continuous therapy is more beneficial to patients than adaptive therapy. Now, this is purely a theoretical model. But the point is that actually when you start to link the outputs of dynamical models to some of the survival models that already exist, where we have already identified what the regression coefficients are in terms of the link between tumor burden and the probability of experiencing an event, what you tend to find is that the optimal strategy is actually continuous therapy. It's not actually intermittent or any sort of other sort of adaptive therapy. But that's a theoretical model, right? So I guess my sort of summary and sort of questions that I have in my mind when I'm thinking about adaptive therapy is that, okay, you've done some interesting studies, but I guess my first question is that given the uncertainty around these biomarkers not fully capturing the treatment effect, you kind of want to see a randomized trial, right? So you'd want to see a well-designed randomized controlled trial exploring adaptive therapy versus continuous dosing. And that's probably a lot harder than you think if you think about the amount of how would you blind patients from this, et cetera, et cetera, et cetera. And what algorithm would you want to use? Another question I guess I have is, why haven't people been using clinically relevant drug concentrations in preclinical experiments? I mean, I think what's interesting is that when you look at the sort of the concentrations that are actually seen in patients, they actually are nowhere near as high as what's been used in preclinical experiments. Another question I have for the community is that clearly there's lots of data analysis and modeling that's been going on outside of this community, which actually have lots of interesting inferences which I've talked about, including this idea that tumor burden is a time-dependent prognostic factor. You don't need to have this idea of a critical tumor volume. People have already built, for example, in the PKPD modeling community, models and provide code to show you how you can actually link tumor burden time series to time to event models. So these things are already there, and you can just use them as they are actually to do your theoretical analysis. I think what's interesting to me is more about understanding, well, how much of the drug effect does a biomarker have to capture such that you feel comfortable making dosing decisions using that biomarker? And there's a public health thing here, right? So in certain situations where actually certain treatments actually don't lead to that higher degree of response rate, is it really in the public health interest to sort of spend so much money on exploring adaptive therapy techniques for a small portion of patients who actually do well on treatment anyway, compared to the rest of the patients who actually don't do well on that therapy at all, and you don't see really much sort of perturbation in their tumor size. So one last quote from Doug Altman. This is what I always think about, actually. So what should we think about researchers who use the wrong techniques, either willfully or in ignorance, use the right techniques wrongly, misinterpret the results, report the results selectively, cite the literature selectively, and draw unjustified conclusions? We should be appalled, yet numerous studies of the medical scientific literature in both general and specialist journals have shown that all of the above phenomena are common. and this is surely a scandal. So I really urge you to go and read some of the stuff that's written in terms of applying models to clinical data from people like Doug Altman. They really do sort of make you think about what you're doing. And finally, I just wanted to promote some useful links. So I've heard quite a few people sort of saying, well, I'd like to work with data, not just in this meeting, but also from, I think, the annual math biology meeting, which I was at earlier this year. And actually, there's a great resource. There's something called Project DataSphere. So if you want to work with individual level patient data from trials, it's free to access. There's tens of thousands of patients worth of data on Project DataSphere. It's really good quality. It's got imaging information. It's got everything you need to be able to start thinking about how you'd apply some of these more theoretical models to the actual raw data. And because I do work for the NHS, I thought it'd be a good idea to advertise. For those of you who are working in the UK, the NHS does actually have like a dedicated website for people interested in mathematical and statistical modeling and general data analysis. It's called the NHSR community, and we run annual conferences, workshops, etc. And they're worth sort of dialing into. You tend to make a lot of the events sort of recordings free for people to watch online. And that's the end of my talk.
Okay, thank you. Great talk. Thank you for sharing diverse view. So we have a couple of several questions for you. So the first question is from Katrina Stankova. So in the case of non-small cell lung cancer on your slide 8, what treatment this concerns? Is it genetic or?
Yeah, so it's essentially what it was. It's what the FDA do. They do this every so often for different tumor types. They'll look over a 10 to 15 year period, look at all the clinical trials that have been submitted to them. So it'll be a mix of all the approved therapies in non-small cell lung cancer. So it'll be a mix of some of the targeted agents that have been improved in lung cancer. So there'll be some of the original sort of targeted agents like gefitinib. So that's Iressa, by the way, crizotinib, etc. So it's a mix of what's been submitted to the FDA over that 10-year period, basically, a 10 to 15-year period. And they do that for other tumor types too. So it gives you an idea of what's happening in terms of what are the general response rates we're seeing in clinical trials, both in the control arm and the treatment arm. It's quite an interesting resource and quite interesting papers.
Okay. And she has a following up question. So what do you propose for prostate cancer instead of a PSA? So
I think circulating tumor cell count. So there was that paper by Howard Scher and Glenn Heller. So they did a lot of work, right? So they worked with the drug that you folks have been working with, abiraterone, and their whole purpose, so Glenn Heller is quite keen on trying to establish surrogacy of biomarkers. So they used, I think, 2 or 3 of the abiraterone phase 3 studies. And what they found actually, circulating tumor cell counts is actually a better surrogate biomarker than PSA. So it fully captures the treatment effect on the endpoint. So if you're going to make treatment decisions, I
think we just lost him. Okay.
I think CTC counts would be the way forward. You're going to use a
I think we have some disconnection, so can you repeat?
Yeah, so sure, yeah. So I said there was these papers by Glenn Heller and co, which are in here, which I referenced. So basically, they've shown that circulating tumor cell counts are probably better in this setting for abiraterone in terms of capturing the treatment effect on the endpoints. If you're going to use a biomarker, it seems like circulating tumor cell counts are the one to use for that specific drug.
Okay, so the next question is from Elsa. So thank you for your great talk. So many important points you addressed. So the link between tumor shrinkies and survivor, is this dependent on approach? Is the data all from treatments that aim to shrink the tumor?
Yeah, so that's what they were. So the papers that I presented, again, are from the FDA. So it's worth looking at some of the analyses that the FDA do. They have an internal sort of mathematical statistical modeling group. And so they release these papers every so often when they have a large population of data. So what they showed there in one of the papers I cited is that essentially tumor shrinkage measured at the first visit, for example, generally has a correlation to survival. However, that link is not drug independent. So in that sense, yeah, you just got to be careful that it does seem to be drug dependent. However, the link does exist.
Okay, so next question is from Indra Jasink. So the question is regarding the result that larger tumors lead to worse result. So what are some mechanistic intuitions to be behind these statistical results?
So I guess it just makes common sense, right? You kind of know that the larger the degree of tumor burden that you have, probably the more aggressive it is, especially in these settings, because these are metastatic settings. And one of the things we do know is that if you start off with large tumors initially, there's higher probability of actually seeing new lesions appear. So again, from a lot of these sorts of results, they do make intuitive sense, I would say.
Okay, so the next question is from Sandy Anderson. So given we know that MTD will drive resistance and treatment failure in metastatic castrate-resistant cancer, metastatic melanoma, especially or metastatic disease. So what do you think should change if not a more evolutionary inspired therapeutic approach?
So I'm not saying that we shouldn't use evolutionary therapeutic approach. I just think you should do the studies well, right? So design them well. So actually do assess these things properly, right? So design, I don't know, I actually think you need a multi-arm clinical trial, right, to really assess this, because you've got different algorithmic approaches, right? So you should think about the design of experiments. Clearly we know that all these therapies lead to resistance of some sort, right? That's the case of everything, even immunotherapies to a degree, right? But I just think we should design these studies better and actually think a lot harder about how you design and are you making them clinically. So the preclinical experiments, are you making them clinically relevant by using the right doses, the right concentrations? Are you using co-culture xenografts, co-culture assays, etc.? Yeah, hopefully that answers part of that question.
Okay, so the question is from Jessica Cunningham. So in the tumor burden studies, where having a large tumor burden result in higher hazard, are these tumors resistant to resistant tumors?
So so essentially those studies are essentially from drug studies, so where you generally see initially a response and then relapse, right? So a lot of those analyses are done in studies where actually you do see resistance emerge. So initially what you do see is that over time, if the tumors shrink, your probability of experiencing an event is quite small. But as soon as the tumors start to grow, depending on the size that you have, that actually does relate to the event. But it is considered, it is in studies where you generally see drugs that have an effect first and then eventually you see relapse.
Okay, so she has following up question. Could high tumor burdens of sensitive cells be less hazard than high tumor burdens of resistant cells?
So you're basically saying, should the hazard coefficient be different for the sensitive component versus the resistant component, I think. So if you do play around with that, which I did with the model, actually you get results that don't necessarily always make sense, right? So what you tend to find then is that actually the drug doesn't work at all, regardless of what you do. So the original trial results actually don't hold if you were to change around the hazard in terms of saying actually the probability of experiencing an event is different between the sensitive population versus the resistant population. And I think Nicola Melillo, the sort of person who I got to help me do some more simulations on this stuff, I think he's going to be writing that up at some point soon. So yeah.
Okay. So the last question is from Joel Brown. So very nice talk. He finds much to agree with your observation.
Hopefully
so do you
think,
think? Quite soon. Yeah. So do you think if it would have been possible to get large phase 3 clinical trial of adaptive therapy without first having the current promising phase 1 trial?
So you've got to be careful of what you call that phase 1 trial. I mean, historically, people do this a lot, right? You do a single arm study, you do some historical comparison and convince yourself. So that's what you call confirmation bias. Now, I think you've got to be careful of that. I think you should do a study, but you should design it properly and not over-egg it, right? So don't overstate the conclusions. I think this is where I think, and I'm not sure if grant bodies help, but we should try not to oversell stuff. Actually just report the results, discuss what they are, and prevent yourself from doing incorrect historical comparisons, right? And then move on, right? So then move on, and rather than doing lots of smaller studies, start thinking about, well, what would that multi-arm final trial look like? Okay, clearly you'll have a control arm, which is on continuous therapy, but are there multiple sorts of algorithms you want to use in that trial to look at adaptive dosing? Yeah, that would be my comment really on that.
Okay, so next question is from Maxi Strober. So based on your assumptions, there is no way for adaptive therapy do better. So do you think there is any benefits to it?
So that was a theoretical model. So one thing we have to be careful of is theoretical models and empirical observations. So I do always try and distinct the two if I can, right? So that theoretical model put me in a bit of a weird situation. So I built that model actually because I was a believer in adaptive therapy actually. but I wanted to see what happens if you link it to a time to event model. And I was a little bit surprised I actually couldn't find a set of parameters that would give me that increase in overall survival with adaptive therapy, right? So then that put me in a difficult situation. So I wanted to sort of support a clinical trial that looked at this, a randomized trial, but I'm kind of in a weird ethical situation. I've actually developed or done some simulations that show that adaptive therapy will never work. So should I really be involved then, or trying to push for a trial that looks at a comparison between continuous therapy versus adaptive therapy? That would be maybe well considered unethical from my point of view to be involved in such a study, because I've already done some simulations that show that it doesn't benefit the patient, right? So why would I subject a patient to an experiment where I've shown myself that it doesn't work? So I would just be very careful about relating theoretical observations to empirical observations. I think what you guys need here is clearly a well-designed, large-scale trial with multiple arms to really get to the bottom of this, I think.
Okay, so here's following up question. So there is a large randomized phase 3 trials of intermittent androgen deprivation treatment, which is shown to be difference in survival between continuous and intermittent arm.
So what I found is that actually there's many studies, and actually some are conflicting to each other. So if you go to, I think there's been a couple of meta-analyses that essentially have shown in the end there is no difference. However, that's intermittent, right? That's not what you want to do. So you got to be really careful of what you confuse in terms of intermittent versus adaptive. And actually, you got to, I think what you should look at actually is there's been multiple studies for intermittent androgen deprivation therapy. And I think has been a couple of meta-analyses. And in fact, I think there's a Cochrane review, which when they did the sort of meta-analysis, they essentially said, well, actually, there is no difference. Yeah.
Okay. And from Yannick, don't you think that your result depend on your modeling choice, like the growth rate function and the form
so that was the point. So the thing here to remember is that dynamical model that is the signal that's most important. So you can play around with that signal. So for example, like I said, this postdoc Nicola Melillo has played around with multiple different parameter options. So essentially he's done a Monte Carlo simulation exercise, and he's never really found one, and I haven't either actually. So it doesn't really matter about this. It's not dependent on the dynamical model. It's the signal, right? So what feeds into these time to event models is the actual signal itself, right? What generates that signal is kind of irrelevant. You could actually just make it up. You could use sine curves if you want to mimic adaptive therapy, and you'd still end up with the same results. So yeah, I mean, I've seen that you guys have published your paper. And if that models and the parameters is there, we could easily hook that up to a time to event model and see what happens, right? That should be straightforward.
Okay, thank you. So we don't have any more questions. So thank you for your talk.
Great,
and
thank you.
Thank all the speakers in the first session. So we will have, so this is the end of the first session. Thank you.